Um, so now it's my pleasure to introduce Dr. Allie Gardner, who's a assistant professor of arthropod vector biology in the School of Biology and Ecology, and also a faculty fellow in the Mitchell Center. I'd forgotten about, even though I met Allie a long time ago, about her background, that she doesn't just have an undergraduate degree in biology, but also in history. And I think maybe it's just me, but I think some of the ways she brings together diverse ways of looking at these issues probably benefited from that way back when she was an undergraduate at Williams College. Then she did 3 different degrees at the University of Illinois in veterinary medical science, in statistics, and her PhD in entomology. And I've been really looking forward to this talk because she's done so much in such a complicated area, bringing together a wonderful team. But I did want to tell you that when I look back at some of the things she said about why she does what she does, something really stood out for me in a faculty profile on the Mitchell Center. And this isn't exactly what she wrote, but it's close, a little bit of a paraphrase. It says, although it's well documented that many human activities influence the transmission of vector-borne disease, there have been limited efforts to translate these findings into practicable disease management strategies. This is due in part to the formidable challenge posed by understanding, and here's the most important part, changing human behavior and priorities. And so my current projects involve collaborations with social scientists to integrate natural science and human dimensions research. I just want to say that like she came to her own view of that, but that's why the Mitchell Center was created, is this idea universities are really good at producing new knowledge, lots of great faculty and students and others doing that work. But there's no guarantee that that knowledge that we produce is actually going to change the individuals and the institutions that is what's required to go in the directions we're trying to go in. And so to me, Allie's work is an excellent example of both the opportunities of that, but also the challenges and why it's complicated and why people like her who really stick with it are key to getting this done. So please join me in welcoming Allie Gardner.
All right. Well, hi, everyone. I'd like to begin by thanking you all for the opportunity to talk with you this afternoon about some research that I've been working on with my colleagues here at UMaine over the past few years. I'm an assistant professor in the School of Biology and Ecology at UMaine, and I've also been a member of the One Health group in the College of Natural Science, Forestry, and Agriculture for the past 6 years that I've been at the university. And I'm a medical entomologist. I'm interested in the ecology and management of vector-borne disease transmission. I'm particularly interested in emerging pathogens and emerging disease vectors. And this is a research area that really has no shortage of interesting challenges to explore because throughout the 21st century, vector-borne disease regularly has been headline news, right? I grew up in the New York City suburbs, and between the persistent threat of Lyme disease, the outbreak of West Nile virus during the early 2000s, pretty much vector-borne diseases all over the place. There's no escaping the persistent threat that it poses to human health. And of course, this is a global problem as well, right? So within the past 20, 30 years, we've seen the emergence not only of Lyme disease and West Nile virus, but we've also seen other mosquito-borne virus epidemics, dengue, chikungunya, Zika viruses. In the area of tick-borne disease in the Northeast, we've been seeing multiple emerging tick-borne disease systems, babesiosis, anaplasmosis, most recently Powassan virus as well. And what we've seen in the news really is backed up by the data. Vector-borne disease currently comprises nearly a third of emerging infectious diseases worldwide. And globally, it's responsible for over 1 billion cases and over 1 million deaths per year. This paper that we're looking at these data from is getting a little dated at this point. This is a 2008 paper, but this trend has really continued up till the present, and there's really no sign that the emergence of vector-borne disease is slowing down. And there are a variety of different factors that contribute to this phenomenon, right? So we know that global travel and trade, for instance, have facilitated the movement of disease vectors and the pathogens they transmit to new locations over the past few decades. We also see that a combination of land use change and climate change have facilitated the establishment of these vectors and pathogens within their introduced ranges. Over-reliance on conventional insecticides has facilitated the evolution of insecticide resistance and the failure of many of our long-standing tactics that we've used to manage vector-borne disease historically. And what I'd really like to point out is that what all of these have in common is that they're related in some fashion to human activities. And this really adds a layer of complexity, I think we can argue, to both understanding the ecological principles that underlie the phenomenon of vector-borne disease emergence and the persistence of vector-borne pathogens. And it also adds a layer of complexity to our development of disease management strategies as well. These are just extraordinarily complex systems that we're looking at here. And so within this context, I've worked on a variety of different vector-borne disease systems. All of my research efforts are thematically linked by three focal research aims, whether we be considering tick-borne disease in New England, dengue, Zika, chikungunya viruses in the Caribbean, West Nile virus as well throughout North America. The three major research themes that I've been working on throughout my career are first, investigating the interacting ecological and social conditions that together enhance arthropod-borne disease transmission. Second, attempting to explain and predict the spread of emerging and reemerging arthropod-borne diseases in the landscape. And then third and last of all, to David's point, one of our major goals in my lab is to actually translate our findings to devise effective and sustainable disease control strategies that are ideally grounded in ecological theory. And so in the course of pursuing all of these collective set of goals, one of the things I've really enjoyed in my career is getting to interact with a very diverse group of colleagues spanning different disciplines. We routinely collaborate not only with other entomologists and disease ecologists, also with data scientists, molecular biologists, economists, social scientists. So we really bring a wide range of disciplines together to understand how human activities and the human environment relationships can facilitate the establishment and persistence of vector-borne disease, as well as look for places where we can break the chain and inhibit disease transmission. So during this seminar, I'm going to focus on one specific interdisciplinary project that I've been working on for the past 3 years that focuses on the Lyme disease system. And I'll use this as an example of some of the approaches we might take both to studying these integrated socio-environmental systems in the context of vector-borne disease, as well as some of the ways forward that we're trying to move in terms of actually changing human behavior to mitigate vector-borne disease exposure risk. And I want to start off by showing some Lyme disease human case data as a way to show the motivation for this problem, to set the stage a little bit. So what we're looking at here in this map is the number of Lyme disease cases per state in 1992, which is the year that Lyme disease became a notifiable illness, meaning that doctors, healthcare providers are required to report cases of Lyme disease to the CDC for surveillance purposes. So you can see here that we've got a good number of cases in 1992, about 10,000 human cases nationally. And then I want to look at the same map 25 years later. Here we are in 2017. And you can see that this map really has shifted. This landscape of Lyme disease transmission in the US really has shifted. For one thing, the number of reported cases has multiplied by 4, right? We're up to about 43,000 reported cases per year by 2017. And I do want to emphasize that this is likely a tenfold underestimate of the true number of cases that occurred in the US during this time. And we also see this dramatic increase in the number of cases in two particular focal areas of tick-borne disease transmission, one of them here in the Northeast and the other in the upper Midwest. You can see over that time period from 1992, when we had like virtually no cases in Maine, now we're one of the higher incidence states for Lyme disease, by 2017, which is the second year that I was at UMaine. And part of the reason why Lyme disease is so challenging to manage is because of the complex life cycle of the black-legged tick, which is the principal vector for Lyme disease here in the Northeast. Ticks have this very complex 2-year life cycle with multiple molts moving from one life stage to the next. So from larva to nymph and from nymph to adult. And each time the tick molts, it needs to take a blood meal, is the entomological term that we use, from a vertebrate host. It needs to parasitize a vertebrate host in order and digest that blood meal in order to be able to advance to the next life stage. And this is really, consequential. This will help set up some of the research questions that we're approaching later on in the talk. For adults, for the larvae and the nymphs, the hosts tend to be like small mammals, things that are close to the ground, right? Like we get a lot of rodents, we get a lot of ground-dwelling birds as some of the key hosts for the juvenile ticks. And many of these small mammals that are hosts for juvenile ticks also are key hosts for the pathogen Borrelia burgdorferi that causes Lyme disease in humans. For the adults, the hosts that we're looking at typically are larger mammals like deer or dogs or people. And although these hosts don't actually typically harbor pathogens that ticks can transmit, they can serve to amplify tick-borne disease risk in the environment by supplying blood meals to large number of ticks and increasing the tick population size. So again, during the juvenile stages, we've got these ticks encountering these small mammals that are likely to harbor pathogens, and then parasitizing more animals that are likely to become ill from pathogens or that could amplify the tick population size later in the life cycle. Now in Maine, we have multiple different diseases spreading that are transmitted by the black-legged tick. This is a map of Lyme disease cases, excuse me, per 100,000 people over the past 5 years. And although Lyme disease is the dominant concern right now in Maine among the tick-borne pathogens that exist here, I really want to emphasize that other pathogens like human babesiosis, human granulocytic anaplasmosis, Powassan virus, also are emerging and also are transmitted by the black-legged tick. The reason that's important is because to me, this really demonstrates the importance of ecological understanding to management of tick-borne disease. Even if we had, for instance, a Lyme disease vaccine, which we currently don't, there are numerous other pathogens that the black-legged tick can carry as well. And so really the only viable way to address all of these threats simultaneously is to develop management strategies that are going to target the vector itself or otherwise seek to kind of disrupt vector-host interactions. Ecologically based management, to my view, could offer a solution that captures exposure risks to multiple tick-borne pathogens simultaneously. And in the state, we also have a very interesting socio-ecological context for tick-borne disease transmission as well, because so much woodland area, especially in the areas of the state with very high Lyme disease incidence, are privately owned. So what we're looking at in the map here, this is a map of the percentage of non-industrial private forest land ownership by county throughout the state of Maine. And you can see that in these southern areas of the state, where tick-borne disease incidence is highest in southern and coastal Maine, over 80% of the forest land actually is privately owned by small family forest landowners that own, in all likelihood, somewhere between 5 and maybe 10,000 acres. And so the situation that we have here, this is really the genesis of the research project that I'm going to discuss, because what we're looking at here is this situation where family landowners are effectively controlling the ecological conditions that might alternatively enhance or inhibit Lyme disease transmission among wildlife, as well as spill over into the human host population. And so it's really critical to understand how these landowners' decisions impact transmission and whether certain management decisions could potentially help to mitigate the problem of Lyme disease transmission in Maine. And so over the past 3 years, I've been leading this project that we call the Maine Forest Tick Survey that investigates the impact of forest management on tick-borne disease transmission from both biophysical as well as social science perspectives. And so this is our team. They really are the most extraordinary group of collaborators that I've had the privilege of working with so far in my career. Two of our co-PIs on the project are Jessica Leahy here in the School of Forest Resources, and Carly Spinarski, who's now with the Canadian Forest Service. She used to be at UMaine as well. Both of them work in the area of human dimensions of natural resources. The other co-PI on the project is Laura Kenefick, who's a silviculturist with the US Forest Service with the Northern Research Station. She's also based out of Old Town. And Alyssa Ballman is the coordinator of the outreach and citizen science components of the project. And we also have a large team of graduate students and postdoctoral researchers and undergraduate research assistants who the undergraduate students aren't pictured here, but nevertheless are a very important element of the team. And so I'm going to start off talking about the biophysical research components of the project that my lab has been leading. I'm going to talk about some of our tick data, and then I'm going to move into the human dimensions research and the citizen science project, which is our attempt to kind of tie all of these strands together with biophysical and social science data collection taking place simultaneously on the same properties. And so over the past few years, my lab has been working to understand how forest management practices might influence tick-borne disease transmission dynamics, and again, whether forest management could even potentially inhibit tick-borne disease. And forest management, I want to emphasize, is a really appealing tactic for tick-borne disease management because it has the potential to affect multiple stages of the tick life cycle, right? So what we're looking at what we're looking for in that complex life cycle of the black-legged tick is ways that we can disrupt the life cycle, ways that we can disrupt interactions between ticks and their hosts that might ultimately result in suppressed disease transmission. And so let's say that we take our forest management practices, right, whether they be like prescribed burns or removal of invasive plants or logging. There are a few different pathways by which these management decisions can affect ticks and their hosts and their interactions. First of all, management practices can really impact the microhabitat features on the forest floor to which ticks are exposed, things like temperature and humidity and leaf litter cover. Ticks are really sensitive about humidity, like most arthropods, due to the surface area to volume ratio. They're really vulnerable to dryness. And so microhabitat features conceivably can alter abundance of ticks in the environment if the ticks aren't able to survive whatever those conditions on the forest floor happen to be. And at the same time, our management practices also can influence wildlife communities. And again, these are the host communities that ticks interact with throughout their life cycle in order to complete those different molts and be able to reproduce. So management practices has been shown in other research to impact things like the diversity of vertebrate hosts in the environment, the density of key hosts for the black-legged tick, like white-footed mice and white-tailed deer. And so collectively, this impact of forest management on wildlife communities can alter tick abundance, right? If we've got more key hosts present in the environment, that could potentially increase the tick population size, and the reverse. And the wildlife communities also influence tick-borne disease infection prevalence. because again, some hosts are better pathogen reservoirs than others. Some are better able to amplify pathogens in the environment than others. And so collectively, tick abundance and then tick-borne pathogen prevalence in the environment collectively add up to what we think of as entomological risk of exposure to tick-borne disease. This is generally the density of infected ticks in the environment are altered by this ecological cascade that's initiated by forest management practices. So again, a very appealing tactic, I would argue, for tick-borne disease management. So one of my lab's current research interests concerns the impact of timber harvesting on tick density. There's been a lot of research already on the impacts of invasive plants and fire on ticks, but timber harvesting is a very common management activity by private woodland owners here in the state, and its impact on tick ecology, I think kind of surprisingly, remains understudied, given its importance to our natural resource economies. So Stephanie Hurd is a PhD student in my lab. I'm going to talk a bit about her thesis project. Her thesis project focuses on tick densities and pathogen infection prevalence in stands that are under kind of different intensities of active forest management through timber harvesting. And so from 2019 to 2020, she led this field study that was based in southern Maine. Again, I'll just put you in mind that this area of the state is important, not only because we have a very high tick-borne disease incidence in this area of the state, but also because we've got this really high proportion of non-industrial land ownership. So she selected these 5 towns, and within each of these towns, she identified 3 experimental units that have variation in the number of trees per acre, as well as the basal area per acre. And so trees per acre is, basically what it sounds like. It's just number of trees per acre. Basal area is the average amount of area that's occupied by tree stems. And so as you can imagine, these are like very correlated measures. You can see this is a figure that shows the trees per acre and basal area for these different experimental units that she was working with throughout the study. The reason why we focused on these measures in particular is because they're standard measures that are used by foresters developing forest management plans for production or regeneration or whatever the landowner's objectives for their own woods might be. The way that we read, like the common tool that's used by foresters are called stocking guides. They basically have these 2 different lines, the A line and the B line. If your basal area and trees per acre fall above the A line, we'll refer to this as an overstock stand. Between the A line and the B line, we've got a fully stocked stand. Below the B line, we've got an understock stand. And so the reason why I'm going into all of this is, again, because one of our key research objectives here is ultimately to be able to make recommendations that landowners could actually use and incorporate into their existing management planning. So we're working really hard here to try to use the language of forestry and use a language that's going to be accessible to the stakeholders that we're ultimately hoping to impact through the work. And over the course of this study, Steph did a variety of different measurements. Again, our goal is to understand the impact of stocking on our tick densities and our tick infection prevalence. So she collected ticks using like a drag sampling technique where you basically just drag a white cloth on the ground and the ticks will attach to the cloth. You can count the ticks. This is regarded as one of the most reliable measures of host-seeking ticks that we use. She also conducted very in-depth forest measurements at multiple levels of the forest structure. She collected trees per acre and basal area data during her initial study site selection. If any foresters are in the audience, she did that using a prism. She measured canopy closure using a densiometer, saplings using fixed radius sampling plots, leaf litter depth and leaf litter cover as well. So all of these variables are things that could conceivably in some fashion interact with the life cycle of ticks and their hosts. And then Steph also took microclimate measurements. She had eye buttons that were positioned at the ground level and some that were positioned at the height at which ticks typically seek hosts. So we could also understand how these forest management practices and the harvesting intensity is going to cascade down to all of these different microhabitat features that could conceivably impact the tick life cycle. And so what we found in this study is that the structure of forest stands really varies widely in response to timber harvesting history. And we were considering harvests that were conducted between 10 and 15 years prior to the study. So these pictures are going to give you a little bit of an idea. We have some stands that have no recent harvest history. So that is, they have very high trees per acre on one end of the spectrum. And then on the other end of the spectrum, we have some stands that were harvested very recently. They tend to have very low trees per acre associated with them, right? We don't see nearly the amount of large trees in the more recently harvested stand compared to the stand that hasn't been harvested within like the past 20, 30 years or so. And of course, this is a spectrum. We see lots, in between these. But this impact of trees per acre has cascading effects like all throughout the different levels of the forest. In our forest with no recent harvest history, again, our high trees per acre stands, we have high canopy closure. So there's going to be very significant shade in the forest understory. We see low saplings and large ground vegetation since they don't have, a whole lot of light to grow. We're going to see a smaller amount of saplings, a smaller amount of large ground vegetation. We do see a high amount of small ground vegetation presence, a reasonably high leaf litter depth as a result of those really closed canopies. And interestingly, we also see a very kind of stable humidity and temperature in these sorts of environments. Not a whole lot of variance in either humidity or temperature over 24-hour periods. And then when we look at our more recently harvested stands with our low trees per acre, we see basically the reverse. We see a lot of sunlight because there isn't very much in the way of canopy closure. We see a lot of saplings and large ground vegetation now that the canopy has been opened up for them to grow. A lower small ground vegetation presence, a lower leaf litter depth as a result of the forest being dominated largely by saplings. And we also saw that there was much higher temperature, which stands to reason because, again, we've got a lot of sunlight filtering down in this environment, and humidity was more variable over a 24-hour cycle. And so what we're interested in, of course, is what is the relationship of all of this to black-legged tick densities? And all of this, as you can imagine, had very strong impacts on black-legged tick densities. These are extremely different environments that we're looking at, and the tick densities were reflected in this. In general, our main observation was that lower trees per area, which again is typically associated with a more recent harvest, tends to reduce densities of nymphal black-legged ticks through impacts on the forest floor and understory. So our main observation again is this positive association between trees per acre and the number of host-seeking ticks that we find. We also used a random forest statistical model to test the associations between various stand characteristics and tick densities, because we're really interested in identifying, what specific feature of these forests is the primary ecological driver of our tick densities. And three of the key associations that we saw here, we saw that small ground vegetation tends to be really strongly associated with densities of ticks. The maximum temperature in the leaf litter, the higher the maximum temperature, the higher the density of ticks we tended to see. And we also saw that there's a certain relative humidity range that seems to be the sweet spot for tick activities. When we saw about like 85% relative humidity, that's when we saw the highest tick densities, our host-seeking ticks through the drag sampling. And so it seems that there is this relationship that exists between our timber harvesting and our tick densities, but that relationship mainly seems to be driven by what's going on in the understory and the forest floor, that environment that the ticks and their hosts are going to be exposed to directly. Now, while these results, mainly are related to vegetation and microclimate, there also is evidence in other areas of the literature that active forest management history can have strong impacts on population sizes and activity patterns of wildlife. So we were also interested in the impacts of this timber harvesting on the wildlife species that are really key to the tick life cycle. We started off looking at the impact of timber harvesting on deer based on trail camera data that Steph had collected, but we didn't detect any relationship, at least initially, between white-tailed deer activity and forest stand characteristics, or for that matter, between deer activity and black-legged tick densities. When it came to the small mammals, we had a couple different hypotheses regarding the mechanistic processes that might be going on here. Our first hypothesis was that timber harvesting impacts mouse population size, directly altering the encounter frequency between ticks and rodents, as well as the density of infected nymphs in the environment. Our second hypothesis, which is a little more complex, is that timber harvesting might have an indirect effect on rodent populations. If we have impacts of harvesting on predators of mice, perhaps the rodents, even if the population size isn't changing at all, perhaps they're going to engage in avoidant behaviors. They perceive fear as a result of perceiving predators nearby. That could in turn alter the tick rodent encounter frequency and the density of infected nymphs, but through like the indirect pathway instead of this reduced rodent activity as opposed to a change in rodent population size. And so in order to address these questions, Steph set up another study where she did a combination of like live trapping rodents to estimate population size. She also conducted some behavioral assays to estimate rodent activity patterns. She used track plates to record rodent activity as well. And then for her live trapped rodents, she also estimated the tick burdens on the rodents, trying to like tie all of this together and understand some of the mechanistic pathways by which the timber harvesting could potentially cascade down to impact tick wildlife interactions. And we're still in the process of analyzing these data, haven't gotten through all of it yet, but one little teaser that I can give based on what we've seen so far in the preliminary data is that 55% of the seed, so she did this behavioral assay measuring rodent foraging behavior and found that 55% of seed was eaten in the high trees per acre stands, whereas only 45% was eaten in the low trees per acre stands. So it could be that this is another mechanism by which the density of nymphs is being impacted as a consequence of timber harvesting. Okay, now all of these studies so far have focused on tick ecology in the summer, but the last point that I want to wrap up on for the biophysical research is that we're also really interested in survival over winter, because the conventional wisdom is that winter temperatures impose a major constraint on the geographic distribution of ticks in the state. And so another master's student of mine who recently graduated, Michelle Volk, conducted a study of tick overwinter survival spanning large-scale ambient temperature and snowfall gradients in Maine. And so at each of these field sites, Michelle set up an experiment where she established 16 enclosures that look like this, and she seeded them with live ticks. And then to each of the enclosures, she applied one of 4 experimental manipulations. She either removed all of the snow cover and the leaf litter cover from these enclosures, or she removed one or the other, or she left both intact as a control, representing like the natural conditions where we do expect to see snowpack and leaf litter offering insulation to the ticks. And what we found in this study is that under both cold conditions up north in Presque Isle and comparatively warm ambient conditions down in Cape Elizabeth, removal of snowpack and or leaf litter significantly reduced overwinter survival of black-legged tick nymphs. And so this is really consequential because we know that forest management practices could alter both leaf litter depth and snowpack in forest habitats. So this represents just yet another pathway by which active forest management potentially could alter black-legged tick ecology and thus our risk of exposure to tick-borne pathogens in humans. Now, so far we've been focused on biophysical aspects of this system, but part of our goal for the Maine Forest Tick Survey was to put all of this ecological research into its social context. So this is a little schematic that we put together illustrating the feedback loop that we expected to see between land use and land cover change and tick densities and tick-borne disease prevalence change. We've already been through the bottom half of this diagram, looking at the impacts of forest management practices on our composition of forest communities and our composition of wildlife communities. But what we're interested in now is a potential feedback, whether this feedback exists, where exposure to tick-borne disease in the environment could potentially change the way landowners think about how they protect themselves and even potentially how they manage our land, which is kind of our ultimate goal for this ongoing research. So the social science elements of this project were designed to measure exactly this. Can exposure to tick-borne disease alter landowner perceptions around forest management and around tick-borne disease, potentially affecting landowner decision-making and feeding back on transmission of tick-borne pathogens in the biophysical system. All right. So we already spoke about how forest management is a very appealing tactic for area-wide tick control because there's potential for it to disrupt multiple parts of the tick life cycle. But it's also appealing, I think we can argue, because there's an opportunity here for tick-borne disease management to complement landowners' other aims for their forests. So this is a landowner typology analysis that was conducted in 2011. And what the authors attempted to do in this study was classify private forest landowners into different groups, depending on what it is that they want to be doing with their forests. And you can see that people have just absolutely diverse objectives for their privately owned land. Some of them are really interested in production and getting some economic benefits from their land. Some are more interested in protection and conservation of either wildlife or forest. Some are recreationists who want to be going out and enjoying, hobbies on their land. Some of them are passive. They just have their land. Maybe it's been passed down in their family for generations. They're not really doing anything in particular with it. They just have the land. And so we're really interested in how does all this different decision making, wind up affecting our tick-borne disease system, and is there a way for the recommendations that we'd make based on the biophysical research to complement what people are already trying to do. All right. So in this phase of the study, in 2019 and 2020, we conducted a large-scale private forest landowner survey to address three major questions. First, we want to know, what do private woodlot owners already know about tick-borne disease? Second, we want to know, do landowners incorporate tick prevention into their woodlot management decisions currently? And then third, and I think the million-dollar question, how willing would they be to adopt management behavior to reduce future tick exposure risk? Is there a willingness to change here as they gain knowledge? And so to address these questions, my colleagues, Jessica Leahy, Carly Spinarski, and our graduate students, Casey Leknowitz and Katie Perry, developed a 16-page questionnaire that they delivered to different landowners in southern Maine in our same focal study area through two different mechanisms. First, part of the surveys were distributed through a drop-off pickup survey design. So here the surveys were administered in person at residences. There were two opportunities for folks to get the filled out surveys picked up in person. And then we had a mail return option as well. This generated a high response rate, like 70% response rate. And then we also distributed the same survey through a mail survey. Here there were 3 mailings. The response rate was considerably lower, around 40%. But you can see that the vast majority of our responses came in through the mail survey. The reason why we have these different numbers is because the mail surveys we distributed to a much larger geographic area, whereas the drop-off pickup surveys were all centered around New Gloucester. And so this was, again, a 16-page questionnaire. The majority of the questions were close-ended 5-point scale questions. And many of the questions were adapted from the National Woodland Owner Survey, as well as other previous work that was concerned with woodland landowner decision-making, as well as Lyme disease prevention. And this was an extensive survey. We won't have time to look at all of the data in depth, but I want to share with you some of the key highlights that we got from this survey research. So one area we're really interested in is, what do private landowners in Maine know about tick-borne disease? And the answer is that by and large, they're pretty knowledgeable. The one of these statements where a low proportion of participants got the answer correct, they actually weren't aware that Maine has the highest rate of new Lyme disease cases per capita right now. Some other more general observations from this survey, just some other general insights. We found that about 20% of private woodland owners had awareness of any forest management strategy that could be aimed at reducing tick densities. Of those strategies, only 6% reported awareness of any single management strategy, which I believe was invasive plant removal. The most important motivating factor for any future changes in land management, this was held by 80% of participants, was reducing personal risk of tick-borne disease exposure. Unsurprisingly, our participants had a very high degree of tick-borne disease exposure. 25% of them had indicated a previous tick-borne disease diagnosis, which is an elevated exposure risk in this group relative to the American population broadly posed. Barriers to action that our participants identified were concerns regarding the time and cost of interventions and access to information. And then we also observed that in general, female private woodland owners had higher levels of tick-borne disease concern compared to average, which is consistent with previous research that seems to suggest female private woodland owners generally hold stronger ecological values. All right, so the last piece of this project that I want to talk about is our citizen science project, which we've been running as well as an effort to tie together some of the different themes from the biophysical research and from the social science research. And so our citizen science project, the Maine Forest Tick Survey, has been led by Allison for the past 3 years. It's an active surveillance citizen science project, the first active surveillance citizen science project around ticks nationally, which means that we're training participants to actually go out and collect ticks on their properties using the same techniques that we use as researchers. We had 261 citizen science participants who completed the study between 2020 and 2021, an 83% retention rate, which we thought was, we were very proud of that result. All of the citizen scientists were located in southern and coastal Maine and owned woodlots greater than 10 acres. In 2021, we allowed between 5 and 10 acres to be participating in this study as well. And again, the idea of this study was that participants dragged for ticks on their own land, and then they completed questionnaires about their forest management histories and decision-making. So just to give you an overview of the training and data collection for the citizen scientists, this involved a Herculean effort on Alyssa's part with this study. We developed 4 different questionnaires that participants filled out in Qualtrics over the lifetime of the study. One of them concerned the participants' motivation to participate in citizen science. We also had a survey, a preliminary survey about tick-borne disease attitudes and beliefs. We asked the participants to go out and measure forest vegetation using a simplified scheme on their own property, so we could get the idea of what their land looks like. And then they answered a questionnaire as well about the forest management history on the property where their tick collections took place. And the citizen scientists were trained through live webinars over Zoom in how to collect ticks on their own properties. And they conducted tick collections 3 times during the month of July. Again, this was active surveillance using the same drag cloth technique that we use in our ecological field research. They completed time collections where they drag for a certain amount of time and then report the number of ticks that they found. And then all of these ticks ultimately were lab tested for 3 tick-borne pathogens, Borrelia burgdorferi, which causes Lyme disease, Babesia microti, which causes babesiosis, Anaplasma phagocytophilum, which causes anaplasmosis. And we gave all the citizen scientists their own tick collecting kit that had, a lot of all the supplies that they needed to complete their collections. And again, just to give you a general idea of what our data looked like from this study, here are the tick counts that we found in 2020 and 2021. You can see that there's a really significant difference in the tick counts between the 2 years, almost more than 5 times the number of certain tick species in year 2 compared to year 1, which is fairly typical for Maine. We have some years that do have much higher tick densities compared to others, which we expect reflects the variation in the small mammal populations from year to year. We also had 440 non-tick specimens that were accidentally submitted by citizen scientists in 2020. We were kind of interested in identifying what some of those mistaken IDs were. And we found a lot of spiders and mites and small hemipterans like leafhoppers, true bugs were very commonly being mistaken for ticks by the citizen scientists. So that's a kind of interesting little side study that we've been working on. Our non-tick specimen identification for 2021 is ongoing. This is our pathogen infection prevalence data for 2020. We just got the data for 2021, and we're still kind of sorting through that. But we found that 25% are infected with the Lyme pathogen, 8% with anaplasma, 6% with babesia. So just to give a general picture of what our disease exposure risk is around the state. And although we're still in the process of analyzing the data, I want to show you just some very brief, interesting teaser results. One of our observations, this is again from the 2020 data, we're still working through 2021, but we found that properties with more recent timber harvests generally had significant fewer ticks compared to those that had been harvested a long time ago. That supported the results of our field studies at a much larger spatial scale. We also found that properties with invasive plants, which included barberry, bush honeysuckle, common and glossy buckthorn, had significantly more ticks compared to properties that didn't have invasive plant species, which also is consistent with prior research. And then on the social science side, this is an interesting observation. We asked participants, how concerned are you about Lyme disease on your property? And the answer is they are concerned. Over 75% of participants are reporting that they're either extremely or moderately concerned. But on the flip side, when we ask them if they're avoiding areas of the property that may have ticks, the answer is definitely not. So this was an interesting observation here that I think speaks to some of our challenges moving ahead as we try to change behavior. There's this mismatch between woodland owner concern about ticks and their willingness to avoid even certain areas of their land due to these risks. So just to wrap up here, I want to leave you with a few conclusions that I've taken away from this project that hopefully can be generalized to other vector-borne disease systems, maybe other environmental problems in general. Ecological and social conditions, right, are clearly major drivers of vector-borne disease transmission, as well as spillover into human populations. We know that multiple pathogens that are transmitted by the same arthropod vectors are emerging. So therefore, I would make the case that vector management offers the most sustainable approach to preventing spillover to humans. And our last point that's kind of guiding our work as we move forward in this system, that we really need collaborations among scientists spanning the disciplines, healthcare professionals, stakeholders, and at-risk communities to devise and execute the long-term solutions to persistence of vector-borne disease that we're looking for. Again, this is kind of where I'll leave the research at this point, but we're really looking forward to continuing collaborations in this group. I feel like we've just started to scratch the surface in terms of the questions that we can ask, as well as the interventions that I think are necessary in this system to change human behavior. I'd like to wrap up again by just acknowledging the many students in my lab who have participated in this research over the years, especially Alyssa for her amazing effort coordinating the citizen science project, as well as, again, collaborators, Jessica, Carly, and Laura, who I've really loved working with over the past few years and look forward to continuing with in the future. And on that note, I think I've got some time to take a few questions.
Can we give a hand to Allie for that terrific talk? So even though she can't see us, she'll know that we're all clapping so much because it was such a great talk. And that we are so fortunate, Allie, to have you as a colleague and faculty member at UMaine, and you've shown us in this talk why that is the case. And David started out saying, you know, here are some things that we need to think about. What does she do? And why does she do it? And that we heard over and over again the very great importance of looking for practical solutions. So really getting a deep understanding of what's going on and understanding that in a way that can lead to practical solutions. And we heard very early that really tick-borne diseases and diseases of this sort are headline news. They're global problems, and that there are a variety of factors that are operating, like world travel, land use change, climate change, insecticide resistance, lots of different things. So we need to produce new knowledge, and she has shown us the ways in which they're producing new knowledge, but asking the question of, well, will it change things? And what's going to be important, and really focusing on those areas where there might be the possibilities of change and what could actually happen. Many people who have been here today have been just really interested in asking questions. Like one of the things, Allie, people were saying, you've helped us understand this is really important, but how does this compare, say, to COVID deaths versus, you know, if we think of COVID versus vector-borne diseases, just to help us kind of think about, you know, what's going on.
So how does this, okay, I'm going to try to interpret. I think that there are a lot of different ways that question can be interpreted.
Sure.
Yes, but so like tick-borne disease and COVID, I would say are kind of different systems, in the sense that with COVID, we're looking at a pathogen that's directly transmitted from human to human. There isn't, you know, quite as strong an interaction with an environmental system involved necessarily as there is in the tick-borne disease system, where this is really all about how the vector is interacting with wildlife and with the environment, and then with us. But that being said, there definitely is a very strong behavioral strand, that human behavioral strand, that's running through both systems. Certainly. And one aspect, I think, of the tick-borne disease system that we haven't even approached in this study is how does like human behavior and interaction with the woods alter human risk of exposure through that mechanism as well. So I do think that there are some parallels here. The transmission mode definitely adds some kind of unique dimensions to each of those pathogens. But I think that, you know, they both are systems that certainly exemplify the need for the role of human behavior in driving the trajectory of disease transmission. And I think on the deeper level, COVID also shows us the potential consequences of zoonotic spillover. Although COVID has become more of a human-specific disease at this point, there's a lot of transmission human to human, unlike what we see in the Lyme disease system. Certainly COVID also is of a zoonotic origin. So we see in the context of COVID what the consequences can be of human encroachment onto wildlife habitat.
Thank you. One of the things, again, of the many interesting things about your talk is you over and over again helped us understand the importance of coming up with hypotheses. So you watched what you learned, you thought about what you learned, you worked with your partners in terms of what you learned. Do you have any insights as future scientists that are in the audience are thinking about coming up with hypotheses. How do you do that?
Sure. Yeah. So I think that a large part of our research always involves like first looking at broad patterns that we see occurring in either the natural environment or in a human system. So our research, before we started exploring like the mechanisms that are underlying all the interesting patterns that we observe, initially, what we're interested in is, going out, measuring things, seeing what's out there and trying to find correlations. Only at that point, we really can start getting into, I think, these deeper questions that I find very exciting about causation. And so I would say to anyone who's, initially developing hypotheses and developing research questions, the main important thing to do is to go out and observe, see what's happening on the ground. And from there, you can begin to pose questions about, okay, well, what's causing this? How can we design an experiment that can like isolate a certain mechanism that we're interested in? And try to develop our understanding of what actually causes these processes to take place.
Thank you. We're almost out of time. There are so many different questions, but let me ask one more, which is, you talked often about different disciplines needing to come together. So if a whole group of students came to you and said, what should I major in? I want to study this. I want to understand this. What should I major in? What would you say? What would you recommend?
Well, that's an interesting question. So I think that one of the things that I've really appreciated in my career is that you definitely, cannot be the expert in everything, no matter how appealing might be. And that's really where the value of collaborations comes in. I've been exposed through my collaborations, and I should mention that I was introduced to my collaborators on this project through the Mitchell Center. David put me in touch with many of them when I first started my position at UMaine. I think one of like the nice things about team research is that you don't need to be like any specific major in order to be able to engage in it. Like we truly need all the viewpoints. And so, yeah, I guess in terms of choosing a major, I would just advise anyone to that ideally in college, we're going to be learning skills about how to write and how to communicate, how to interact with people from different viewpoints, regardless of which particular subject area we're studying. And the major is simply the context for the development of those other skills. And so I would say to gravitate toward an area that you're really interested in in-depth reading in, that you'd like to imagine as kind of the intellectual context in which you're going to be working. But to my mind, the goal of the undergraduate education is not at all about like facts or information per se, so much as learning about how to have a flexible mindset and how to interact with people who bring like diverse intellectual bent to the same question.
That's wonderful. That's just such great advice for people. And we, again, all so appreciate all that you've said, and there are many questions that people added in the last few minutes. So I hope you'll have a chance to look to them as well. We all look forward to following your career.
Well, thank you so much for the opportunity and for these great questions.
Thanks, Allie.
Thank you.